On to question 8. We're nearly through the paper. Thank you. Lyme disease is most frequently caused by the bacterium Borrelia burgdorferi. Borrelia burgdorferi. Lyme disease can be difficult to diagnose. Figure 5 shows an ELISA test that is used to find out if a person has antibodies to B. burgdorferi. Okay, so ELISA test, that's what is that? Enzyme-linked immunosorbent assay. So the main thing to remember is that it's enzyme and linked. That's the EL, because then you'll know that an enzyme is involved. And here they've explained the ELISA test they've carried out. Step 1, B. burgdorferi antigens are added to a test well in a dish. So they've added the antigens of the bacteria into a dish. They then take a sample of the patient's blood, it's added to the well. If the B. burgdorferi antibodies are present, they bind to the antigens. So essentially, you've got a well, you've got the antigens of the B. burgdorferi. They then add the patient's blood. If the patient's blood contains the antibodies for the burgdorferi, then this happens. Okay, now a scientist looking at this under a microscope cannot tell this has happened. So what do? Well, we then wash it. The well is washed, right? Now, if none of these antibodies attach and the person is completely clear, then it's going to look back like this. There'll be none of these black antibodies I've drawn. After it's washed, these will all wash off. But if they're attached, they won't wash off. They then add a second antibody with an enzyme attached. This binds to the B. burgdorferi antibodies if they are present. So and this is actually called indirect ELISA. When you use a second enzyme, it's indirect ELISA. Okay. If you just use one antibody, sorry, when you use a second antibody, it's indirect ELISA. I said enzyme there. So what they do now is they're like, right, how can we prove that this situation has happened? The person has it. It's a positive result. That would be a negative result. But how can we prove this has happened? Well, what they do is they add a second enzyme, and this second enzyme attaches, is complementary, so forms an antibody-antibody complex. This is an antibody-antigen complex. This is an antibody-antibody complex. And the second enzyme normally has a colored bead. It might go purple. It might go blue. Think of the COVID tests, pregnancy tests, blue. So it has a colored bead. Oh, sorry. It has this enzyme. Actually, they don't use color beads in this. What am I on about? They don't have color beads at all. It just has an enzyme attached. So this is an enzyme linked, hence the EL in ELISA. This is the enzyme linked antibody. So what's going to happen now? This will still be colorless. We still don't know if the person has it. We're going to wash it again. We're going to pour water through it again. Now, what that means is if this is the situation and there were none of these, right? If the person was negative, then these have nothing to connect to. So when you wash it, they all disappear and you end up like that. You're finally, in the final step, you're going to add a substrate, which is converted to a different product if the enzyme is present. So now you add the substrate. If you add a substrate to this, because you've washed it, and nothing attached, then the substrate will not change color. However, if it's like that, when you add the substrate, you're going to get like a color change because the substrate will react with the enzyme. The enzyme is present and you get a color change and you say, sorry, sir or miss, you have B. burgdorferi. Anywho, let's zing on. And that was just a brief explanation. I have got other videos. You can check them out here on the ELISA test. Right, let's get on with the questions. A false positive in this test is a result which correctly indicates that antibodies B. burgdorferi are present, which incorrectly, oh my good gosh, I want to get that wrong. So a false positive indicates that they're present when they're not actually present. A failure to thoroughly wash the well in step 4 can result in a false positive. So the false positive would be like this. The person, here are the antigens in the well. The burgdorferi antibody isn't there, but then they add the enzyme, which is supposed to attach to the other antibody, but the other antibody isn't there. So they add the enzyme link one. They forget to wash it. Now, when they add the substrate, the substrate changes color. And we get some sort of color change. And we go, oh, they've got it. But of course, we forgot to wash it. If we'd have washed it with water, all of these would have disappeared and we wouldn't have got a color change. So what we need to do is put that in words. We need to say, because the enzyme linked antibodies are still present So when substrate is added, we get a color change. So you should only get that after washing, which proves that the enzyme link 1 is attached to the other antibody. Anyhow, that will get you to fat bad boy marks. Bitch bash. Now let's move on. So going down here to 8.2, a false positive can be produced if a person has been infected by another bacterium that causes the disease called syphilis. So you can get a false positive as well when you don't really have it. And that will be that when they put the antigens for B. Burgdorferi, the person has actually got the disease syphilis, and syphilis's antibodies have a similar 3D shape. So they form an antibody and antigen complex with the B. Burgdorferi, even though these are syphilis antibodies. Okay, so what we want to say is the syphilis antibodies have a similar 3D shape or tertiary structure, so form an antibody dash antigen complex, right? So basically, yeah, there we go. Let's zing on. 8.3, a false negative. This is where they say, congratulations, you don't have B. Burgdorferi, but you actually do have the B. Burgdorferi. A false negative is often produced if the person tested within 2 weeks of being infected with B. Burgdorferi, of course. So they're looking, do you have the antibodies? Do you have the antibodies? If you have the antibodies, you've had an immune response. This means you've got B. Burgdorferi. But if they do it after only 2 weeks, what has your body not had time to do? Your body has not had time to produce the antibodies, okay? Or you don't have enough of the antibodies. After only 2 weeks of a primary response, remember, this is the first time you've been infected with it, so your memory cells haven't produced any. After only 2 weeks of a primary response, you would not have enough antibodies. I shouldn't say you would not have, you may not have, would be better. You may not have enough antibodies. What the hell is an antibody? You may not have, I guess, some sort of walking tree creature. Ents. Oh, that's Lord of the Rings. After 2 weeks of a primary response, you may not have enough antibodies. And what we're talking about there is, do you remember, like, with it goes like that, primary response, bosh. If you've got the memory cells and stuff, then yeah, you're gonna have like loads of antibodies in your system. So this is only the after effects of a primary response. And so yeah, you might actually be infected. We could say you have not produced B plasma cells yet. Not enough B plasma cells cloned, not AB. Every time I type, my fat fingers hit other keys. Not enough B plasma cells cloned. Not enough memory cells cloned. Yeah, or differentiated. You haven't made enough of those, so you're yet at that point. Okay. Jeez Louise, this looks disgusting. Sometimes the symptoms of Lyme disease can persist for 6 months following antibiotic treatment. This condition is known as post-treatment Lyme disease syndrome, PTLDS. Scientists investigated the symptoms experienced by a large number of PTLDS patients and a control group during a 2-week period. They asked all the participants if they experienced symptoms of PTLDS and to record the intensity, the strength of the symptoms. The scientists used a statistical test to determine if there was a difference in the intensity of the symptoms between PTLDS, between the two groups. Probably like a t-test, right? To see if there's a difference in these ratings. This would be a t-test. Table 3 shows some scientist results, including the probability values obtained by the statistical test. The scientists concluded that more PTLDS patients, scientists concluded that more PTLDS patients than the control group experienced symptoms and greater intensity of symptoms. Evaluate, is this true? So I'm going to write my answer on either side of this, and we kind of want like, yes, it's true, and kind of, no, it might not be. Okay, so evaluate is always things for and things against. So straight away, you've got to understand what these P numbers mean. When the arrow is like that, this means there is less than a 0.001 chance that the difference in fatigue was down to chance, just luck, or just randomness. Okay, so there's a 99.9 whatever percent chance that there is a significant difference in fatigue felt. And there's highly significant difference in joint pain, fatigue, and muscle pain. So I'm going to say highly significant difference in joint pain, fatigue, and muscle pain, um, less than 0.001, right? P. And then I might want to also say a significant difference in depression and fever, um, lower significance than above. Yeah, so basically, the patients declared that there wasn't as big a difference in the intensity with those. There's a much higher significant difference in the fatigue, the joint pain, and muscle pain that the PTLDS group feel. Okay, so there's some points there that will probably pick me up some marks because we've talked about this that they've given us. We can say that the percentage, we can also just make the general statement, right? What's the general statement? Well, look, 100 to 57, 96 to 32, 40 to 4, 35 to 3, 86 to 62. The percentage of PTLDS group they experienced more intense symptoms. So the PTLDS groups uh, percentage, PTLDS group percentage experiencing symptoms higher than control on all symptoms. I should say PTLDS group experience intensity, or yeah. The intensity of the symptom was higher in all. So I should say PTLDS group intensity of symptom. Oh God, I think I'm just messing this one up. I'm trying to say the intensity of the symptoms for all of the symptoms, the intensity. Oh, that's much better. Let's write it like that. For all of the symptoms, the intensity was higher in PTLDS group. No wonder these are so difficult to word these when they have all this fat information, but that's biology for you. So I've done at least three over here. What can we say about this? I think it was done only over 2 weeks. So we can say it was only done over 2 weeks. Also, the scientists what else can we say? Let's check it out. Intensity. When someone says, "How tired do you feel?" Rank it on 1 to 5. Or, "How much pain is your joint in?" Rank it from 1 to 3. What do we call that? Where you're asking a patient to basically tell you, and how much you've gone to the doctors probably, and how much would you say from 1 to 10 that your headache hurts? So it's subjective. So the intensity of pain is subjective. And what we mean by that is it's down to the patient to say, yeah, that really hurts, or no, that doesn't hurt. It's not like they tried to measure impulse activity in certain pain sensors or something, which would be more of a quantitative, objective way to actually measure the pain. Okay, which is probably really difficult to do. So that's going to cover us all for the evaluate. Just trying to think of anything else. What was the size of the group? Oh, well, I don't think they told us the size of the groups. We don't know the size of the control group. We know it was only done over 2 weeks. I'm gonna leave it at that. All right, guys, that's the end of that question. Okay, question 9 is about the ELISA test. Oh my days. Dengue fever is a human disease caused by the dengue virus. Scientists designed an ELISA test to detect antibodies to the dengue virus in a patient's blood sample. Figure 10 shows a diagram of this test and some information about how it works. We got diffusion from X of the anti-human antibody with the enzyme attached. Essentially, that's just a monoclonal antibody coming down from X, and it has an enzyme attached that will react with a substrate and create a color change if you have the disease. And from Y, we've got the patient's blood sample diffusing upwards. Figure 11 shows the negative and positive results that were produced 20 minutes after the use of the test shown in figure 10. So we can see the negative one, you get a control line, but if you're positive, you get a control and a test line. So guess what is on the test line T that creates the positive result? So what do we think is stuck on test line T? We're going to need a color change, right? And if you need a color change with an enzyme, what must be present? We know that the enzyme linked antibody is moving down, the ELISA antibody is moving down from the circle at the top. So what must be present on the T when it gets there? that it changes color. And it's going to have to be a substrate because the enzyme is attached to this antibody. That's my little Pac-Man enzyme there, attached to the antibody, and it diffuses down towards that line T. And on the line T will be a substrate. So if it gets there and it binds with the antigen, the immobilized antigen or whatever's there, not the immobilized antigen, but if it binds with an antigen that's in the patient's blood and it reaches that line and binds with the patient's blood, the timing will have to be right, then it is going to change color, that line. And it's going to bind with the antigen, and there's a substrate attached which will lock into the active site of the enzyme and be catalyzed and change color. So we need a color change. We need a substrate to bind with the ELISA enzyme and cause the color change. Also, the antigen that is complementary to the antibody has to be present, and that will get you 2 fat marks. So let's put that down here. The antigen must be present on line T to bind with the monoclonal antibody to form an antigen-antibody complex, but also we must have a substrate present on line T because it's the substrate that will lock into the active site of the ELISA enzyme and create the color change that allows us to know we have a positive result by lighting up line T, purple, pink, blue, whatever the color change is. All right, let's zing on to 9.2. A line at C shows the test has worked. Suggest one reason why a line at C shows the test has worked. We'll look back up. We've got the ELISA enzyme diffusing down, and we also have the patient's blood diffusing up. If we get a line at C, what does this tell us? This tells us the patient's blood has made it past the T test area. And that means that it didn't contain any antigens if it got to C, and it just proves that it's at least gone past the test area, because you might not get a line on T and you might think, great, I'm not positive for dengue fever, but it might just be that the blood sample hasn't got to T. So by having a line at C that lights up when the blood reaches it, we know that the test has worked and the person doesn't have dengue fever. Right, figure 12 shows a flow chart for the anti-human antibodies, with basically how to make monoclonal antibodies. You did this in GCSE if you did AQA, and you should know this off by heart. So it says, suggests why the cells, the fused cells, allow continuous lots of production of monoclonal antibodies. Well, do you remember about the hybridoma at GCSE? That's the fused cell here. You don't have to call it hybridoma. But do you remember what it's made from? It was made from a cancer cell which rapidly divide by uncontrolled mitosis, and it was fused with a B lymphocyte from a mouse. In this case, we've got a rabbit, so it's fused with a lymphocyte, which can produce the correct specific antibody that you want to make lots of. Okay, so here we want to make the antibody so it was complementary to the antigens on the dengue fever virus, but that's neither here nor there. Your 2 marks are for saying cancer cell, uncontrolled mitosis, fused with a B cell that can produce antibodies. Yeah, ethics peoples, evaluate the ethics of this. So what can we say? You're injecting rabbits, you're harming rabbits. So something about it being not good because you're harming rabbits. And then that's our negative thing about the production process, is that you harm rabbits. What's the positive? You make lots of monoclonal antibodies and they're used to save lives with diagnostic testing in medicine, to find out if people have HIV, to find out if people are pregnant, to find out if people have dengue fever, people have COVID. So actually, there are really good benefits for medicine and the medical industry for diagnostic testing. And monoclonal antibodies aren't just used in tests like this dengue fever test. They're also used in like targeting cancer cells in chemotherapy and stuff to deliver the chemicals, so they only attach to the mutated cancer cells by binding to the antigens, and they carry a drug and that kind of thing. So basically, we just want to say that they're really good to be used in medicine. They can have lots of benefits, but at the same time, there's an ethical question about cruelty to animals. 2.1, what is a monoclonal antibody? So that's the first part of the question. What's a monoclonal antibody? Well, these are identical antibodies. How do we say that in A-level language? Well, what is identical about the antibody? From one antibody to another, what is it that is identical? It's the tertiary structure, so the 3-dimensional shape of the antibody that's identical. So they will all bind with the exact same antigen, a bit like a lock and key theory. the antibodies are all complementary to the same antigen. So how do we word that? We say monoclonal antibodies are antibodies with identical 3D or tertiary structures. Now, another definition of monoclonal antibodies is that they are all produced by clones or identical plasma B cells. So all these antibodies are made by plasma B cells or clones, identical cells producing the exact same antibody. Don't say something like monoclonal antibodies have the same DNA. They don't have DNA or genes. They're actually just a string of polypeptide. They're just some polypeptide chains, aren't they? They're just a sequence of amino acids, but they have the identical sequence, so the identical tertiary structure. Now, the second question, 2.2, after a disease is diagnosed, monoclonal antibodies are used in some medical treatments. Give one example of using monoclonal antibodies in a medical treatment. So, if you had lung cancer, some of the cells in the lungs would be cancer cells. Okay. Now, they would have antigens that may be slightly different to the other cancer cells. If you wanted to target them with a drug, with a chemical that kills those cancer cells, you can attach that drug to a monoclonal antibody that is complementary only to the antigen of the cancer cell. So it's very specific in the cells it targets. So monoclonal antibodies can be used to carry medicines or drugs to specific cells. You might have a disease of the liver and you want this medicine to only go to the liver and attach to liver cells and affect those. Well, you would then use a monoclonal antibody that is complementary to the antigens on liver cells. Here I'm drawing a little cancer cell. So let's draw a normal healthy cell. This will be a healthy cell. So I'm going to put an H in the middle for healthy cell in our lungs. And then next to it, I'm going to draw a cancer cell. Now, because of the mutations, etc., it's got slightly different antigens to the healthy cell. This is great because this means I can take a drug that will kill and destroy cells, and I will only deliver it to those cancer cells and not the healthy cell next door. So I would attach a drug, which I'm going to use a green to attach a drug to these monoclonal antibodies, these pink ones here. They will only attach to the cancer cell because the antibody is only complementary to the antigen on the cancer cell. The tertiary structure is only complementary, so will only bind to the cancer cell. Those little green squiggles, those are my artistic impression of a cell-killing drug. Well, let's get down to the main question here. 2.3, describe the role of antibodies in producing a positive result in an ELISA test or ELISA test. How do you say that? Anywho, the E in the ELISA test stands for enzyme. The L stands for linked. Sorry, I just realized my microphone was sounding a bit tinny there. The audio was a bit messed up. Hopefully I've fixed that now. So the E stands for enzyme and the L for link. So there's a little clue there. The ISA stands for immunosorbent assay, but you don't need to worry about that. Just try and remember the E is enzyme and the L is for link. Then you'll have a bit of a clue, a trigger to get you started. Now there's two types of ELISA test, and these are really good at searching to find out if you have a certain type of disease. We have the direct ELISA test and the indirect ELISA test. I'm going to go through the direct ELISA test first of all. Okay, so with the direct ELISA test, you can take some sort of beaker or some sort of well, and you're going to immobilize some antibodies, monoclonal antibodies, at the bottom of the well. These monoclonal antibodies are complementary to the antigen we're looking for. So imagine we're searching for some sort of flu virus and we know what the antigen is. We can make monoclonal antibodies that will bind to the antigen. We immobilize those monoclonal antibodies at the bottom of a well. Then we take the patient's blood. We only really want the blood plasma because it's clear. And we can use a centrifuge to get that. You don't need to know that. But we add the patient's blood plasma. Now, let's pretend these little purple diamonds, these are the antigens to the virus we're searching for, or the bacteria we're searching for. These antigens will bind to the immobilized monoclonal antibodies. Now, in between each stage here, or some of the stages here, we really need to wash. And what do I mean by washing? Well, washing is going to remove any antigens or antibodies in the blood plasma that are not complementary to the immobilized monoclonal antibody. So it's important that you wash so you don't get any false positives or any inaccurate test results. So if the antigens are not complementary, they will be removed, and later on that's going to help us find out the person doesn't have the illness or doesn't have the antigen, the target antigen we're searching for in their blood plasma. So what's the next step of this test? Let's pretend they do have the antigen and they're all bound to the immobilized antibodies. Now what we're going to do, we're going to get another enzyme. This is the second enzyme. And in both the direct and indirect tests, we're looking at the direct first of all, but in both tests, the second enzyme is the one that has an sorry, the second antibody is the one that has the enzyme linked. So it's always the second antibody you add that has an enzyme attached to it. So the second antibody with the enzyme attached is added. Now this enzyme is, sorry, this antibody that I'm drawing in green here is a Y shape with the little enzyme like a Pac-Man on the end. This antibody is also complementary to that target antigen, and it will bind to it. Now, we wash it again because this is the most important washing. We need to wash this beaker or this well again because if that purple diamond, that antigen is not present, the second set of enzymes will wash away and will only be left with the immobilized antibodies, right? That's very important that we wash it again because we don't want a false positive. Um, so what we're going to do here now, let's pretend again that the antigen was present. So the second enzyme with the enzyme link has attached as so, like I'm drawing here with the little enzymes at the end. The second antibody, also complementary to the antigen, has attached. What we're going to do now, what do enzymes react with? We're going to add a substrate. Now that substrate is going to have a complementary shape to the tertiary structure of the active site of the enzyme. They're going to fit like a lock and key. They could bring that in if it's a paper 3 question. And the substrate will catalyze that reaction, weaken the bonds and break the substrate. And this substrate will change color and give us a color change. And then we will know, aha, the person had the target antigen. They are infected with that disease. So we're looking for this target antigen. Potentially it's from a disease that we're testing to see if the patient has the disease. And if we get a color change when the substrate binds to the enzyme that's attached to the second antibody, boom, we know they have the disease. So I'm going to summarize this direct ELISA test into just 4 key bullet points. So summarizing the stages of the direct ELISA test. Number 1, the target antigen is going to be added from the patient's blood, if present, to the immobilized antibodies. Because they're complementary, they're going to bind together. Step 2, the second antibody is added with the enzyme link. This will also bind to the target antigen. Then we're going to wash it. In case they don't have the antigen, everything else will be removed except the immobilized antibody, and we won't get a color change. But if they do have the antigen, in the 4th step, when we add the substrate, it will bind in the active site of the enzyme. It will be catalyzed, the substrate, and it will give us a color change, proving that the target antigen was present in the patient's blood plasma. And that is the summarized direct ELISA test. We're now going to look at the indirect ELISA test and what that looks for and what the differences are. Now, first, a quick reminder why it's important for step 3 to do the washing here. If you wash the sample, if the person doesn't have the antigen, i.e. they don't have the disease you're checking for, then the enzyme will be removed when you wash, and then you won't get a color change. You'll know they don't have the disease. If you forget to wash in between step 3 and 4, then the enzyme will be there regardless of whether they had the antigen for the disease. And when you add the substrate, you'll get a color change and you'll get a false positive where you go, you have the disease, but they don't really. All right, let's move on. Okay, the key difference with the indirect ELISA test. So the key difference with the indirect ELISA test is that we are searching for the presence of an antibody produced by the patient's immune system in response to the target antigen. We're not actually searching for the antigen. So this might be a virus where it's really hard to find the virus. It's so small or something like that. And actually, it's much easier to look for the antibodies because a patient who has the disease will produce large amounts of the antibodies. So it might be a lot easier. And this is what they do with HIV when they're testing you for HIV. They actually look for the presence of an antibody in the patient's blood. So they start, instead of immobilized monoclonal antibodies, they start with immobilized antigens. We then add the first antibody that is complementary to the HIV antigen. Now, if you have the HIV antibody in your blood, it probably means you've got HIV, and that HIV antigen will bind, will bind to the immobilized antigen. Now we wash it to remove any antibodies that haven't bound, and if the person is HIV free, then they won't have any antibodies bind to the antigen. So we do a washing here. The next step is to add the second antibody, and just like in direct ELISA, what you have to remember, because it will be on all the mark schemes, the second antibody is the one with the enzyme attached, the enzyme linked antibody. So we add that, and now, unlike in direct ELISA, it's not complementary to the antigen, it's complementary to the first antibody. So you can see here the green enzyme linked second antibody I've just drawn binds to the first antibody that's bound to the immobilized antigen, and then we add a substrate. If there's a color change, the person has it. Of course, we wash again before this. We wash it to remove unlinked second enzyme antibody. But after the washing, if there is any enzyme linked antibody left, then the substrate will change color, proving that that antibody is present for HIV. Okay, final question, peeps. Read the following passage. Kidney cells produce a glycoprotein hormone called erythropoietin, or erythropoietin, whatever you want to say. An EPO molecule contains 165 amino acids and approximately 60% of the mass of a carbohydrate. The EPO is transported in the blood, stimulates the bone marrow to produce red cells. In this way, the red blood cells are produced to maintain the blood's oxygen carrying capacity. Some athletes are basically cheaters and they use EPO by injecting it into their body. The athletic authorities have a program of drug testing to detect the athletes who have injected EPO. In this program, an ELISA test is administered. That's the enzyme linked test. So what we're going to look here is 9.1. Kidney cells produce a glycoprotein called erythropoietin, EPO. Identify two organelles in the kidney that enable the production of EPO. So what can make proteins? What would you make? What organelles would make proteins? Well, you've got the ribosome, which is going to, or the rough endoplasmic reticulum, which is going to construct the protein. It's where translation occurs. And then it folds inside the rough endoplasmic reticulum. But in the Golgi apparatus or Golgi body, we package and modify that. So those are the two organelles you want to mention. Explain the biological advantage to athletes of injecting EPO. Well, if you inject EPO, it told us that you produce more red blood cells from your bone marrow. If you have more red blood cells, you have more hemoglobin, which means you can pick up a greater volume of oxygen, and therefore you can do a higher rate of respiration, more ATP produced, more muscle contraction, more ability to do aerobic exercise for longer and not use lactate fermentation, i.e. anaerobic respiration, which produces lactic acid and tires or fatigues the muscles. So any two of all of that. 9.3. Describe how mice injected with EPO produce an anti-human EPO antibody. Line 14. So how does injecting mice with human EPO produce an anti-human EPO antibody? So you've got to think, you've got phagocytosis occurring in the mice. And during phagocytosis, the white blood cells of the mice engulf the human EPO that's been injected to them. They see it as a foreign invading particle, and they recognize the antigen as foreign. They engulf it, they digest it, and then they present the antigens on their body. They become APCs. Now, the mice also, just like humans, have T helper cells, THCs, and these have specific binding sites or protein receptor sites that bind to the antigen, specific one, and that's when you have the whole clonal selection on the APC, and then that T helper cell will stimulate B cells to differentiate, to clone themselves and divide into like memory cells and plasma cells. And those plasma cells will make an antibody that is complementary to that human EPO antigen. And that's how we make monoclonal antibodies. We can now take that B cell. I mean, actually, really, they take that B cell, don't they? They make a hybridoma by cloning it with a tumor cell just so they can make lots and lots and lots of copies of the monoclonal antibody. But it's not asking us about that. So that's all your mark. So during the ELISA test, what is the role of the anti-human EPO antibody? Well, that is to bind to the EPO antigen. Now, this is part of what we call the indirect ELISA test. This is slightly more complicated than the direct ELISA test. I've got a video you can check out here with a question which will go through it, and probably better than I'm about to do it now. So to bind to any antigen or EPO in the sample in the well. What is the role of the anti-mouse antibody with the enzyme attached? Well, this antibody actually binds to the antibody that's already attached to the antigen. So this antibody is complementary to the human antibody, the human anti-human antibody, and that binds to that. And it also contains an enzyme which will change color with a substrate. Okay, so just to explain here, what I'm doing is I'm drawing the antigens. So they bind these, immobilize these to the bottom of a plastic well. Okay, so like the bottom of a container, these are just immobilized and stuck there. Then they put the sample in there of the athlete's blood. Now, if it contains the anti-human antibody, it's going to bind to the antigen, and we know those are little cheats, but we need to prove it. We need a color change. So what we do is we add a second antibody. Now, this antibody recognizes the first antibody and attaches to that, and this antibody has an enzyme linked to it. Okay, now the green antibody with the enzyme linked to it does not attach to the antigen. It will only attach to the antibody that the human has for the EPO, so which proves that they've got EPO in their system. So it's really important that you do washings with the ELISA test as well, because when you add the substrate, if you previously have washed it and the person wasn't guilty, they had a negative result, then there wouldn't be any of the antibody in them, and then you wouldn't get a color change, and you know that that's negative. So what happens here is this person is a cheat. So when we add the substrate, you're going to get a color change because that enzyme linked antibody, the green one, will not have washed out when you wash it with water. It will have stuck there because it will be stuck to the pink antibody that's stuck to the antigen at the bottom. However, if we had this situation, have a look here. This person is a not guilty athlete. Therefore, when you wash that out, the second enzyme linked antibody will not be present. And so when you add the substrate, the enzyme's not present, so you don't get a color change. That's a negative result. They haven't been cheating with EPO. And if that makes any sense, you are a genius. Anyway, these are your answers here. 9.5. Some people object to the use of monoclonal antibodies in testing programs. Use the information. Well, why are they going to object? Because they tested it, or well, they used mice to produce the antibodies and they injected it into goat. Sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks.
You're still here? It's over.